can start the meeting. So we have Kevin Mizes today with us. He did PhD at Pense Olveshki lab in Harvard. He graduated recently in August 23. And now he's doing a postdoc in Christopher Harvey's lab at Harvard Medical School. And today he's going to talk about the role of the striatum in sequential movements, visually guided or automatic. He recently published an article about that in Nature Neuroscience. So thanks, Kevin, for being with us today.
Yeah, of course. Yeah, thanks for the invitation. And I'm excited to share the work I did with Benza on sensory motor striatum and flexible and automatic motor sequences. Okay, so there we go. 1 second. So our ability to execute a rich and diverse set of behaviors is underlined by the brain's ability to sequence elementary movements in actions in novel ways. And so I like to introduce this, taking, for example, the concert pianist. When provided with sheet music, she can perform any number of sonatas or etudes. But such flexible performance can be mentally taxing and error-prone. And so to prepare for a concert where precision is of essence, she extensively practices a single piece, overtraining it into a consolidated automatic sequence. And although these movements are constructed from the same basic elements, they feel qualitatively quite different to us. For example, it's a lot harder to start in the middle of an automatic piano performance and easier just to start over at the beginning. And this is something you yourself may experience on a more ubiquitous task, typing in your password. So at least for me, I have to delete the entire thing if I mess up in the middle. Whereas a flexible typing task like a CAPTCHA to prove you're not a robot, it's a lot easier to fix single element mistakes. And so in general, we believe that these two types of motor sequences, we call flexible and automatic, engage different parts of our motor system, despite using the same basic motor elements. But the neural mechanism behind this is not abundantly clear. And so to test this distinction, what we wanted a motor task where, like the concert pianist, the same movement sequence can be performed in both a flexible and automatic way. And while we can learn a lot from watching the concert pianist to invasively probe the brain, it's a lot easier to do this in rats. And just briefly, why do we use rats to study our diverse motor behavior? I think they're a good model because they share a lot of the same basic structure, mammalian brain architecture for motor control. Rats are capable of a wide variety of complex motor behaviors, and they're amenable to these invasive experimental manipulations. And so, with these in mind, therefore, taking inspiration from our concert pianist, we set out to develop a piano playing task for rats. And so in this task, rats are trained to press one of 3 separate levers akin to piano keys. The lever press is cued by a light above each lever, just like sheet music, and rats can quickly learn to associate a lever press with this visual cue. And after learning this association, cues can be presented sequentially so that the rat performs a piece of 3 lever presses. And I've been calling this the piano playing task, but more generally in the field, this is a discrete sequence production task. And so having established this, we can then train rats to perform sequences in both a flexible and an automatic way. And so in flexible training sessions, rats can perform sequences of key presses, either cued sequentially by lights, as I just described, or they could do them from working memory, where the second and third press are uncued, and rats must remember and repeat the previously cued trial. And blocks of cued and working memory trials are interleaved and changed throughout the session. Then on a separate session, the automatic session, one sequence is chosen and fixed for the rest of the rat's life. The sequence is overtrained night after night for months over thousands of trials until performed automatically. So here's one rat learning this task. On the y-axis, it's the fraction of correct trials. And on the x-axis is training time in months. And so over the course of many months, this rat can learn to do this task, where each dot is performance in the cued, working memory, or overtrained sequences. And each dot is performance on one session. And we can look from early to late in learning and find that this rat improves the number of correct trials she's performing. And that across many rats, this performance goes up. Rats get better at this task throughout training. And also the time it takes for them to do a trial, measured from the first press to the third press, decreases. both signatures consistent with motor learning. And also, what's interesting is on average, the overtrained sequence was performed both faster and more accurately than the same sequence in the flexible context, consistent with our expectations of automaticity from human studies. But despite these behavioral differences, the basic movement kinematics ended up looking qualitatively quite similar. So I'll show you 3 example trials. And so on the left is one trial of a rat doing the sequence following visual cues, the green lights, which are just those green lights above the lever. In the middle is the same sequence now performed from working memory. And then to compare on the right is the same sequence in the automatic session. And so, whether following the cues or, from memory by repeating the previous trial, or done very automatically, qualitatively, the sequences look quite similar. But we can quantify this. And so to quantify this, we can take the video from a side camera pointed at the levers from the side, and we can track her forelimb as she performs this task. And so this is one rat's forelimb position from 8 example trials in the cued context that I'm overlaying on top of each other and aligning to the first lever press, which is that little blue triangle. And so what's nice to see is from trial to trial to trial, the kinematics look pretty similar, which is something else you might expect throughout motor learning. The movements get more stereotyped. But we can also look at the kinematic traces in the working memory task, and we can look at the kinematic traces in the overtrained task. And in addition to looking pretty stereotyped from trial to trial, we can overlay them and see that across these contexts, the movements look pretty stereotyped as well. And we can quantify this by computing the average trial to trial correlation and see that it's pretty high across these execution modes and across rats. Within a rat, the kinematics are highly correlated. This is consistent across rats. Okay, so now that we've established this piano playing task in which a similar movement is performed in both a flexible and an automatic context, we can interrogate how the motor system differentiates these types of movements at the neural circuit level. And so the motor system is large and distributed throughout the brain. And in general, we assume that it's organized hierarchically with different regions contributing to different aspects of movements. And in particular, flexible and automatic movements are thought to be differentiated at the level of the basal ganglia, and specifically the sensory motor arm of the basal ganglia, which we refer to as the dorsolateral striatum, or DLS. But the exact role that the DLS plays in motor control is debated. And in general, there's 2 major competing hypotheses about what the DLS does in the motor hierarchy, which I'll demonstrate in the schematic. And so one hypothesis proposes that the DLS plays a role in high-level action selection. And so action selection circuits determine which action is to be performed and convey this decision to motor control circuits. And so if DLS was an action selector, we might expect flexible and automatic representation to differ, as the flexible movement would be selected at the element level, left, then center, then right, while the automatic movement, which has been trained and consolidated into a single movement sequence, should be selected at the sequential level, left, center, right. The other hypothesis instead suggests that DLS acts as a low-level motor controller. And so motor control circuits take selected action and they elaborate it into a dynamical pattern of muscle activity to generate an appropriate motor output. And in this case, flexible and automatic movements, which use this, similar kinematics, should look similar at the level of the DLS. And so there's conflicting evidence for DLS both as an action selector or as a motor controller. But these studies typically look at single and flexible sequences where it's difficult to dissociate the kinematics from the sequential level features. But here we can arbitrate between these hypotheses by directly comparing neural activity during the flexible and automatic tasks. And so we implanted tetrodes into the rat's dorsolateral striatum and recorded single neurons from animals as they performed this piano task. And so here's that experiment. So here's a video of an animal performing 4 trials of the sequence in the cued context while I'm simultaneously tracking her forelimb trajectory and plotting spikes from a single unit. And I'm temporarily going to focus on the sensory guided and automatic tasks, putting aside the working memory task for now. And so, as you can see, as she's performing the cued sequences, I'm also dropping these dots every time a lever is being pressed. But we can see across these trials, this unit appears to fire between the second and the third lever press, indicating it's sensitive to some aspect of the movement. But, we're curious how this unit responds then as the animal performs the same movement sequence, but in the overtrained context. And so, thanks to some really nice infrastructure built in the Olveschki lab, we can track this unit over sessions and return to it in the night as she's doing this automatically. And so now here are 4 trials of the animal performing the same sequence in the automatic context. And if this unit were to distinguish flexible and automatic movements, we might expect its spiking pattern to look different across these 2 contexts. But as you can see, she performs these trials in the automatic context. A very similar firing pattern emerges, where this neuron still fires between the second and the third lever press, sensitive to some aspect of the movement, but insensitive to the particular context. And so we can zoom out and look at more trials from other units. And so here's another unit, and we can see it across trials. This trend continues. And so this is just a raster plot where the x-axis is time in the sequence, the y-axis is individual trials, and these small black dots are when the animal presses a lever, and the colored lines are when the neuron fires. And so this neuron appears to be sensitive to some aspect of the movement, firing between the first and the second lever press, but insensitive to the context. This pattern looks very similar across these 2 execution conditions. Here's another neuron. This neuron fires every time the lever is being pressed, but again, insensitive to the cue overtrained context. And here's a third neuron, a little more complicated firing pattern, but remarkably similar across the 2 execution conditions. And so we can zoom out even further and look across the population of all the recorded neurons. And so here's a heat map of the 579 neurons we were able to record. The x-axis is time in the sequence. Now I've warped them to the lever taps denoted by these vertical red lines. And each line on the y-axis is an individual unit, which I've just averaged all its trial activity into one line. And the color map represents the z-scored firing rate. And so across the sequence, there's many neurons that are tuned to some feature of the movement sequence. But what's cool is we can look at these exact same neurons in this exact same sorted order in the overtrained context, and a remarkably similar firing pattern emerges. And so not only are these same units active across the execution context, but they're tuned to similar phases of the task.
I have a question. This is Jonathan Michaels talking, since you can't see me. You present the cued trials are also in blocks. So you present like the same cued sequence for a certain number of trials, and then a working memory one for a certain number of trials. And since the kinematics are quite similar, how do you know that the animal's actually using the cue at all? I mean, why wouldn't they just, do the first cued sequence in a block and then, treat it as a working memory block internally, since they know that they're going to get the same trial over and over? Another way of saying this is if you could look at just the first trial of a cued block, would you still see what you're showing up here, or would it look different?
That's a great question. So there are some behavioral evidence that, even the second and the third trials in the cued task are being performed slightly different than the working memory trials, these behavioral signatures of accuracy, trial time. But we have actually looked at the very first cued trial as well. I apologize, I don't have a figure prepared in the slideshow, but things look very similar if you restrict your analysis to just the first cued trial. But yeah, that's a good question.
Another question?
Is there reaction time difference in between cued trial versus a working memory trial? Maybe I assume that working memory trial may be a little bit longer reaction time than cued.
Sorry, I'm having trouble hearing over Zoom. One more.
Well, that's why I was surprised, because you said the kinematics were so similar, and I would expect that a cued trial would have to be longer because they have to wait.
Yeah.
They can't just assume what's coming. There should be a reaction.
Yeah, rats are so the light turns on instantly. So as soon as the lever press is triggered, the light immediately changes. And so the rats are always in motion. I tried to copy these primate studies where they would, delay the cue and then make the monkey wait to look for these anticipatory movements that are a really nice signature of automaticity, or like, learned movements as opposed to randomly cued ones. And the rats would never wait. So they're always in motion. It is true that if you look at the trajectory correlations, they're a little lower in the cued task. And so there is some signatures that the very first cued trial, is less accurate than the second and the third, and is performed a bit more slowly. But it is remarkable if we kind of warp these things out, you get these remarkably similar patterns, regardless of the fact that this is, the first. cued trial. And another caveat is we are taking the first successful cued trial. And so there are cases where they try to do the first cued trial and they mess up and then it gets reset. And then the counter starts again. And so there are lots of, little caveats to this, but yes, sorry. I hope that addresses the question.
Yeah, thank you.
Yes, and working memory trials also have this remarkably similar firing pattern. And finally, a similar firing pattern across the execution contexts, is apparent in the low-level principal components of the neural activity as well. And here I'm just showing you the that activity evolving over a trial. And so we find that DLS encodes flexible and automatic movements quite similarly. And so while this suggests that DLS is more consistent with a role of a low-level motor controller, it does not fully clarify the coding scheme in the DLS. And DLS may still be acting as a high-level action selector. In particular, it may still be encoding these high-level features such as the lever identity, if you're pressing the left, center, right lever, or the ordinal position, if you're in the first, second, third position in the sequence, which are unchanged across the flexible and automatic conditions. And so to determine, whether DLS is reflecting these high-level features of the sequence, or whether it is truly a low-level motor controller, it's difficult to do this when looking at a single inflexible sequence where these two features are intrinsically linked. And so to disambiguate, we expanded our analysis to include all 12 motor sequences generated in the flexible condition. And so again, here's a heat map of trial average neural activity for the visually cued sequence, left, center, left. Again, the color map is just the z-scored firing rates, where I've sorted them based on the peak firing time. The y-axis is individual units, and the x-axis is time in the sequence aligned to the lever presses. And so now, if DLS is encoding high-level sequential or ordinal information, we might expect the neural representation to look different when the same lever press and orienting movements are performed in different sequential contexts. And so in this case, this means, neural activity representing the left to center movement in the sequence left, center, left should look different if, it is the left center movement in the sequence center, left, center, where it differs in the sequential context and ordinal position. But if DLS acted as a low-level motor controller, tracking only the ongoing, moment-to-moment kinematics, activity should look similar across these two conditions. And so we can look at neural activity, the same units in the same sorted order, and you can see that, remarkably, the same neurons are firing in, the same left center movement, just kind of shifted in a phase in the sequence. And you can see this with the center left movement as well. Really independent of the ordinal position or the sequential context. We can further test this hypothesis, whether DLS is acting as a low-level motor controller, by comparing the cued sequence left, center, left to the cued sequence center, right, center. And so these sequences are composed of different elements. They share the center lever press, but it's in different ordinal positions at the high-level sequential, context. But if we consider these movements in the low-level motor control, realm, in these egocentric movements, they're composed of similar movements. You press the lever, you move to the right, you press the lever, you move to the left, and you press the lever. And so if DLS is acting as a low-level motor controller and is not sensitive to these high-level aspects of the flexible sequence, a neural activity should look very similar across these two sequences, which is exactly what we ended up seeing. These same neurons in the exact same sorted order, and you get a remarkably similar firing pattern that emerges across these two, sequences composed of different lever presses in different orders. This phenomenon is pretty apparent in the neural PC principal component trajectories as well, and holds true over many different flexible sequences. So if we look at these trajectories, for all our sequences, we can see that anytime a rat moves to the right, whether it's from the center to the right lever, the left to the center, the left to the right lever, we can see in our neural PC trajectories, activity follows, this one path down along PC1. And anytime the animal's moving to the left, from the right to the left, the center to the left, the right to the center, activity will follow this path up along the third principal component. And this holds true across, many different sequences. Anytime the animal is moving to the right, activity follows this one path along, the first principal component. And anytime the animal is moving to the left, activity follows this path along the third principal component. And just to quantify this, we can also explicitly quantify it by looking at the correlation coefficient of neural activity across different sequences, for these motor elements specifically. And so just as a control, we can compare on a held out set of trials in the sequence right, left, center, split the trials in two, compute the correlation of neural activity to itself, find that it's pretty highly correlated. We can do this across different sequences then, look at the right to the approach to the left lever press across two completely different sequences, but they share ordinal positions and they share lever presses. Neural activity is very highly correlated. Neural activity is still highly correlated, even if we shift the ordinal position in the sequence, and even if we shift which lever is being pressed, so long as the approach direction is the same. It's only when the animal's approaching the lever from different directions, or pressing different levers and approaching from different directions, does this correlation break down. And this is consistent across cued and working memory trials as well. And so just to briefly summarize, we did not find evidence that the DLS differentiated flexible and automatic movements. And additionally, we did not find aspects of high-level ordinal or sequential information encoded here, but instead found this egocentric movement representation, which was consistent with this low-level kinematic role for DLS, just tracking the tap, move left, tap, move right, tap. And this is even though they've been practicing the same particular sequence for months over thousands and thousands of trials. Rat never developed this consolidated motor representation in the DLS beyond this basic egocentric representation. And so while the recordings, hint at this low-level role, it does not establish the causal contributions of the DLS to motor control. And so, for example, the neural activity in DLS could just be reflecting the ongoing movement, as opposed to actively shaping and controlling the detailed kinematics. And so to test this hypothesis, we performed excitotoxic lesions of the dorsolateral striatum in expertly trained rats and observed what happens to the behavior. And so again, to assess the causal role of dorsolateral striatum on flexible and automatic performance, we can track their performance prior to and following bilateral lesions of the dorsolateral striatum. And so here's a plot of the fraction of successful trials in each execution condition on the 7 days prior to the lesion. So green is for overtrained, orange is for cued, blue is for working memory. And on day 0, the animal is lesioned and then undergoes a recovery period. So she rests in the box and recovers. And then she's returned to the task on day 1. And we can see, what happens to her performance right as she gets back into the task. And so, and then we can summarize this across all rats with these bar plots on the right. And so in summary of a vital role for controlling these automatic movements, which are fully specified in terms of low-level kinematics, performance in the overtrained condition dropped significantly across all rats following the lesion. Interestingly, memory-guided motor sequences, which must be internally driven, were also sensitive to the DLS lesion. But in stark contrast, the externally cued trials were far more resilient to the lesion, being performed with a pretty high success rate immediately following the bilateral lesion. And so this indicates that the DLS is required, for the ability to do this high-level sequential structure of the automatic and working memory task, but is, partially dispensable for the cued sequences, which can be rescued by these external cues. But this addresses the animal's ability to perform these 3-lever sequences. What happens to the detailed kinematics? And so interestingly, we observe motor impairments across all execution modes, not just the overtrained. And so the trial time measured from the 1st to the 3rd lever press increases across all 3 execution modes following the lesion. And we can further examine how the detailed task performance changes by looking at the tracked forelimb kinematics of the active forelimb. So this is just one example animal, and this is 8 trials plotted from the same motor sequence, sampled from the median trial time. And so before the lesion, I've shown you the movements look pretty stereotyped, but after the lesion, the stereotypy decreases following the loss of the DLS. And we can quantify this by computing the average trial-to-trial correlation for trials before the lesion and after the lesion. And so taken together, this suggests that the striatum plays an essential role in specifying the task-refined kinematics independent of the execution mode, but it is dispensable when flexible sequences are rescued by these external cues. Suggesting there is a way for these external cues to guide the basic movement, even though it's with not as clean and refined and stereotyped kinematics, but it is able to guide the animals to perform these flexible sequences. And what's interesting is this sort of encapsulates findings from Parkinsonian patients who experience something called paradoxical kinesia, where they're unable to perform internally driven movements, but when provided with external cues, they can do much, much better. And so here's just an example video of this. So here's a Parkinsonian patient receiving a walking rehabilitation technique using a visually cued technique. And so this is her baseline gait. It's pretty arrhythmic. She's struggling to take her steps. But in the next clip, you'll see the same patient when provided with a visual stimulation. And so this is just circles projected onto the treadmill. And you can see already there's a marked improvement in her gait with these external visual cues. And you can even buy this for yourself at home. Here's another video of a Parkinsonian patient. Oops. Who's able to walk with these external visual cues. And so to briefly summarize, we found that the dorsolateral striatum was primarily not distinguishing flexible from automatic execution and was, most consistently acting as a low-level motor controller. And we further found that although it is required to control the task-refined movement kinematics, it was dispensable in flexible sequencing, rescued by these external visual cues. And this suggests that the action selector has a way to directly control these movements without requiring the dorsolateral striatum. And so I have one more little story for you, which is, one outstanding question we were left with was where in the motor system was the action selector? What was responsible for instructing these flexible cued sequences? And so one hypothesis we had was that it was, there was another subdivision of the basal ganglia known as the associative or dorsomedial striatum that could be contributing to this. And so we'd previously been focusing on the sensory motor striatum due to its known role in movement execution and its large inputs from motor cortex. Whereas in contrast, dorsomedial striatum receives cortical input primarily from prefrontal and posterior parietal cortex regions associated with more flexible cue-driven behavior. And additionally, the DMS has previously been implicated in the flexible control of behavior, such as in the modifying, switching, and updating of behavioral choices in response to previously learned associations. But whether it actually plays a role in our task in this flexible cue or working memory guided motor sequences was not known. And so to test this hypothesis, we similarly performed bilateral lesions in a separate cohort of expert animals. And so here's this result. So here's a plot of the fraction of successful trials for this cohort prior to the lesion in the 7 days. And again, on day 0, the animal has the surgery done and then recovers in a box before returning to the task. And so we had some hypotheses. And so from previous results, we suspected that dorsomedial striatum was not contributing to automatic performance, which can be, is controlled by the dorsolateral striatum. But whether it played a role in controlling these flexibly driven sequences was unclear. And so post-lesion, we found that despite this, this initial drop in performance, consistent with expectations, dorsomedial striatum was not required for performance of the automatic well-trained sequence. But interestingly, it wasn't required for the cue or working memory performance, as much either, with animals recovering within a few days post-lesion. And furthermore, it didn't affect the low-level kinematics. We can look similarly at metrics like the trial duration. We can look at the tracked forelimb position and find, it was unaffected by these lesions, which we can quantify with the forelimb correlation across these trials. And so, this is nice because it's consistent with our previous work. DMS was not required for specifying the detailed kinematics of an automatic motor sequence or of any of these well-trained motor sequences, reinforcing a dissociation between the DLS and DMS in low-level control. But it is interesting that it was not necessary for performing this, visually guided or memory guided motor sequences, at least in this task. And it's likely the DMS is contributing in other ways to this sort of task, such as in early motor learning when gaining this cue lever association, which remains the subject of ongoing work in the Olveshki lab. Okay, and to summarize, in this paper, we developed a motor sequencing task for rodents to address the differences in flexible and automatic execution. We found that dorsolateral striatum, encoded flexible and automatic sequences similarly. and that it encoded low-level aspects of discrete motor sequences and not high-level aspects of the ordinal position or the lever identity. Furthermore, it was required for the task-refined kinematics, regardless of the execution mode, which, we also found that it was required for the automatic and memory-guided performance, but it was dispensable for cue-guided sequences. And the DMS was not required for these automatic or flexible sequences. And this did leave this question still open, what is helping guide these cued sequences without the DLS? Where is the action selector? And we have some ideas, and I'm just going to quickly plug this other paper we have out on bioRxiv, which starts to look into these sorts of questions and also find some other interesting interactions between these types of execution. And I'd like to thank, of course, Ben Solveski for guiding, overseeing, shaping this work, and our collaborators at Columbia, who I did not get to talk about their contributions, but they had some really interesting neural network modeling and suggested some mechanisms. And I'd also like to thank the rest of the Solveski lab for, an immense amount of training and advice on this research. And thanks for your attention.
Right. Thanks, Kevin. Questions?
One question. So after learning the DLS, you showed that the accuracy of the automatic trials were decreased a lot, right? So what kind of errors they were making? Was the error, for example, going for the right target but not pressing it correctly, or going completely for wrong targets in this case? Because if
so we have like a rough analysis on this, and so we've done this error mode analysis where basically I sat down and watched a lot of videos, and many of the errors in the automatic case initially were actually these motor errors where the animal did orient towards the correct lever and did try to swing at it, but just missed the lever or didn't press it all the way. And this was, a dominant error mode prior to the lesion. But after lesion, when I looked at these videos, there were way more of these errors where they're actually going and pressing the incorrect lever. And it wasn't just that they still knew the sequence, they just couldn't physically reach and press the lever. They were actively reaching and pressing incorrect levers. And this gets complicated. The error modes are not this simple. There's all these complications of they get off from like how the box is coding what the trial is to what the rat thinks the trial is. And so sometimes they're out of sync and they're violating the ITI, this intertrial interval. So I just want to clarify that our motor errors and sequence errors don't capture the full scope of errors, but we definitely saw cases where rats went and pressed the incorrect lever.
Isn't that like a sign for maybe this DLS has a role in action selection, or if I understood correctly?
What we think is happening is that they've consolidated this at a low, they've consolidated the DLS sequence in terms of the kinematics, and now there's nothing there, and they're guessing. They've reverted to what they're doing in early learning, where they're just kind of guessing and pressing levers early. And it is difficult to completely dissociate low-level kinematics from these high-level sequencing.
Okay,
yeah, but that's a good point. That's a good point. It is difficult to fully make this dissociation.
I can ask a question if you can hear me.
Yes.
Yes. So I know it's really difficult, and maybe I didn't catch it, but like, what if have you looked at like the activity of the DLS over the learning? Like, so for example, like early in their learning, does that, like, I know the percentage of the correct trial is way smaller, but like, if you look into the correct trial, does anything specifically change in the DLS over the learning between like automatic and flexible sequences?
I haven't looked over learning. So this is just so these rats have been training at this task for a while when we implant them. And so the idea would be if something, diverged when they started learning the overtrained task and became more automatic. But since we're already in such late stage, I don't know if we'd see anything if we recorded over learning and compared. I mean, we haven't done the recordings though, so I don't know for certain.
Thank you.
So, Kevin, really nice talk. I'm puzzled, quite honestly. You're on, by the way. I'm puzzled. So the encoding results, I mean, the firing rates that you show and the tuning functions, I mean, that seems to contradict the lesion results you're getting, right? Where they seem to be you know, if DLS is really just doing low-level motor control, then it's not clear why the overtrained sequences are so selectively affected. And so, do you have recordings in other brain areas just to compare? I mean, like what happens if you record an M1? What do you see there?
We have recordings in motor cortex.
Yeah.
I think maybe I wasn't completely clear. It's complicated. We don't think DLS is controlling everything in kinematics, but it's adding these task refined, so what's learned on top of it. And so you can actually go after these lesions and look at just the single lever presses themselves, and they're not as much affected by the lesion, the shape of the kinematics of just the lever press. If you zoom in just around the lever press. And so we think that's like the role of lower level motor controllers. And what we think it's doing is it's, helping refine these smooth kinematics beyond these traces.
Yeah, I wonder if there are, I mean, I wonder if DLS does have some sequence encoding and some dependencies, but a lot of like maybe 90% of the neural state space is driven by these individual movements as you show in your analysis. I mean, it can be explained by they going left, they going right. But on top of that, there might be some small modulation. I mean, 10% of neurons might be enough to really give you some sequence depending encoding, right? Have you been nice to kind of either, I don't know if you've tried to analyze this in a more quantitative way to see if there's any evidence.
I think it's like very difficult to know if there's not a small population of neurons for sure doing these things. And I have some nice, maybe I can show you the slides. Where we record from motor cortex and find some pretty marked differences. And in motor cortex, we think it's much more consistent, doing things more related to high level action selection and tracking the lever identity. So the big, maybe I can do this. Yeah, so the caveat with these motor cortex recordings is there's not a ton of neurons. It was really hard to get these recordings because we need to record over weeks, and motor cortex is really dorsal and you get a lot of torque. But at least in motor cortex, you can find, here's 3 example neurons. If you guys can see my screen, okay?
Yeah.
Yeah, so there's 3 example neurons that are, sensitive to some aspect of the high level features. Like this neuron is firing for the center lever press and only when it's first in the sequence and not for other lever presses. This neuron in the middle is firing for left to right movements. And then this neuron at the end is, what is that? Oh, this is an egocentric neuron. I think this is a better example. This neuron at the end kind of ramps up over the sequence, and we think it's sensitive of, ordinal positioning. And I ran this classification analysis where you throw things through GLM to classify them, whether they're sensitive to these low level aspects or to ordinal position or to lever identity. And you get way more in motor cortex that are sensitive to these high level aspects. And you do get some baseline in DLS that get detected by this GLM as sensitive to these contexts. And I've gone back and tried to look at these neurons, and it's nothing like as obvious as these neuron 1, neuron 2, neuron 3 here. And what I'm skeptical of what I think it is it's sensitive to something in the kinematics, some subtle difference in the kinematics that shows up across like these different, they do the second to third lever press quicker than the first to the second. And so if a neuron is very, very sensitive to that aspect of the task, it might show up as sensitive to the ordinal positioning. And we just get lost in the noise, and and we can't completely rule out that there aren't these neurons that exist. For sure.
When you say non in your classification, that means that like your first category, which accounts for 50% of your DLS neurons, what does non mean?
None is it's not doing better than the null model, which is just some average firing rate over the task.
Okay.
Yeah, sorry, this was, I apologize, this was rushed through.
So untuned to the task.
Yeah, untuned.
Thanks, Kevin. I'm also have a question, if that's okay. So I was a bit puzzled by the 20% accuracy of the sequence execution for the automatic ones after the DLS lesion, right? So just like the simulation results, I was assuming that if DLS is really fine-tuning those automatic sequences or even controlling them fully, then they shouldn't be able to execute the sequences at all for automatic ones. But you still see after 7 days, there's like, what is that, like 20% or something accuracy? How do you explain that?
Yeah, they're slightly above chance. I think their behavior, if you compare their behavior to just a model where they're randomly pressing, they're certainly doing better than that. What we have is there's kind of a catch in the training. And so when their performance hits chance performance, I add a cue back in. And so this was originally implemented when they were training, as sort of a catch to help them train in the automatic task, to not like remove the cues way too fast and they forget everything. And what happened is they hit this threshold and they started getting cues back in. And so I exclude these cued trials, but there is still some level of instruction being put back into the system that may be, sort of affecting these. And so that's what I think is happening is they have some structure they're trying to relearn with whatever, flexible action selector is left in place. And then they still have these motor control circuits. They still have their brainstem control circuits that, enable them to go perform these lever presses. And so I think, even without the striatum, which is controlling this refined kinematics, they can still do slightly better than chance. But what what we chose to focus on was this, pretty substantial drop that didn't recover. And then the fact that their kinematics, were way more screwed up post lesion and way more variable. So even though there's still so much brain left intact that I think could be contributing to this above chance performance, but I don't know for certain exactly what it is, or if
it becomes more like a working memory task because they get like little reminders potentially.
Yeah. And these cues don't they're not there all the time but it's the rats are kind of riding this threshold that I've set, and like dipping into it, dipping out of it. And
thank you.
If I turned it off, they just would eventually quit, is my guess.
Yeah, thank you.
I have one question. So I think, is it doable with the human subjects and to see any distinction, dissociation between anterior versus posterior striatum, see the similar effects, like as you report here. Maybe you can design some human fMRI experiment with normal people, normal subject with and patient with Parkinson's. Maybe we can design fMRI experiment like that. Do you think it's a good idea to do that kind of experiment?
I mean, I'd be curious though. I think this behavioral task and everything here is very, very heavily inspired by human work, and I know there's I don't know in Parkinson patients, but I think Julian Doyen has some work looking at striatum across these sorts of tasks and finds dissociations between the associative part and the sensory motor part. So yeah, and there's been these also these pretty big monkey studies where they found early late learning, it's more consolidated versus when you're newly acquiring a sequence between the medial and or between the associative and the sensory motor part. So I don't doubt, my instinct would be that you would find something distinguishing these two, but there's also lots of caveats across when you compare rats to humans, that like if you did these lesions in humans, it would, you wouldn't be moving very much, whereas the rats can still press the levers. So that's a major caveat of trying to compare across species.
I can ask a follow-up question on that. Just as you mentioned, like lesioning M1, for example, would completely make you paralyzed, but in humans. But like other papers in Bence's lab, they also show that even if you lesion M1, rats can still do cue-driven movements. So I was wondering, in light of that, what is the area that actually is controlling these cue-driven movements? Because so far you've shown that M1, DLS, and DMS are not controlling them in a sense in the level of selection. So
well, we think We think M1 is controlling these Q-driven movements, and when we've lesioned M1 in this exact task, you see the Q task, drop to close to chance levels.
I see. So
we think it's either M1 or something relies on M1, going through M1.
okay, thank you.
May I ask a question? Do you see any difference after lesioning DLS between your working memory and complete visually guided task?
Yeah, so it's harder to tell because performance isn't as good in the working memory task, but we did see a drop that came with a significant in working memory and went to about chance levels when we lesioned DLS. Whereas the queued task didn't face this drop. And so I've been making this distinction between flexible and automatic movements, but I think there's this other distinction of internally generated and externally queued sequences. And at least with the DLS lesions, it tends to fall along the lines of that dissociation, where the internally generated movements are both messed up, but the externally queued movements were fine, or more fine, more resilient.
Thank you.
I have one more question. It's kind of probably basic and maybe I didn't catch it, but you told that the kinematics between the automatic and the flexible ones were the same, but I think you also told that in the automatic trials, the rats were faster and more accurate. I mean the faster part. They were faster. So by similar kinematics, do you mean only trajectories, like XY positions? So what about the speed here? Speed is also part of the kinematic, right? And if they are faster, so the speed is different and it should be reflected in the DLS, as you said, because it's coding the low level features of the execution, right?
Yep. I think this is totally right. So I think the overtrained movements are faster, but there's a distribution, and the spreads between the speed of the overtrained movements and the queued movements overlap pretty substantially. Though on average, the overtrained movements are faster. And the analyses I've shown you, where we look at these correlations, we're warping to some template length, and so this is distorting, the encoding scheme of speed in striatum, for sure. And the issue is, how do things scale with speed? And for sure, we can find differences that do relate to speed. But on average, when you warp things, they end up, the shape looks pretty similar, and the neural activity looks similar. And then there's certainly effects of speed that we are kind of brushing over here, unfortunately. And the issue was we were trying to sub-select, trials within the same speed to control for this, but we are so trial poor here because you have to get the same neuron across days, across sequences, and that just left us with so little data to work with. And so it was an unfortunate choice we had to make. But I mean, I think I can even show you in an example neuron. You can, maybe these neurons demonstrate it. Yeah, I'm not sure. It's interesting because if you look at neuron 1, there's some variability between the first and the second lever tap. And at least this neuron speed doesn't seem, it has this same little firing burst in between these. Maybe it's not being captured here, but there's other neurons for sure we can find where you kind of get these more stretch speed pull effects.
Yeah, thank you.
Can I ask a question?
Yeah, go for it.
Hi, Kevin. Thanks for the talk. I have a question about the plots where you have different sequences and you have different units on top of each other. The average activity. You have one example here. But you get two of them together that you compare, I think CLC and CRC next to each other to show that the left movement is coded in these group of neurons in both cases.
This plot?
Yeah.
I apologize.
I have a question about almost the half of your neurons in the bottom of the bottom rows, the bottom half rows, the ones that get activated after the sequence is finished. So those neurons, I guess there are two options. Either they decode the task, the sequence being finished, or they decode the kinematics of going backwards, because I assume that after the sequence is finished, Mars goes back to the center, right? So now, considering that about the plots that you had, that you said half of the neurons are at the non-decoding level, I think maybe those neurons are just encoding the kinematics of going backwards, which is also kinematics and is task related, and it's not like at the non-level. Maybe just wanted to like see if you have any thoughts on that.
Yeah, absolutely. It's as soon as the rats get the reward, they get a little tone and they drop down to the little reward spout and they lick for water. And if you watch them, even that action of dropping to the reward spout and looking for water becomes super stereotyped. And I totally agree with you. I totally think these neurons are encoding for that move back, drop down, lick water. And we can actually, this is a kind of broad analysis, but these two plots on the left, I apologize, this is less clean. Oh God. This is on the left is the sequence left, right, center, and then water reward. And on the right is the sequence left, right, center, but it wasn't rewarded. It wasn't what the rat was supposed to do. And you see the rat didn't drop down and drink water, and you don't get that activity. It most likely just hung out, waiting for the next trial to start.
Right, so almost all neurons are encoding kinematics, but half of them are encoding going to the water way, like from different levers that you have. Is it right?
Yeah, I can't answer you for sure because we haven't actually, performed any analyses of the kinematics as the rat drops to the reward port, but that would be consistent with our hypotheses.
Thanks.
Yeah.
Any other questions? Yes, no? Okay. Then thanks a lot, Kevin.
Yeah, thank you.
Yeah, thanks, Kevin.
Thank you.
Thank you. And
Yeah, have a
Thank you
good
so
week.
much.
Yeah, it's fun.
bye.
Bye-bye.